pernicious anemia, a megaloblastic anemia characterized by abnormally large blood cells, results in symptoms ranging from fatigue to numbness of the extremities to death if left untreated. In 1926, George Richard Monod reported the disease was effectively treatable with a special diet, most notably including very high daily liver intake, and the discovery earned him the 1934 Nobel Prize in Medicine. As it turns out, liver is high in cobalt, chelated in the form of vitamin B12, also called cobalamin. Indeed, the anemia is ultimately a disease of cobalamin deficiency. Cobalt, atomic number 27, is a first-row transition metal and essential micronutrient in humans and animals, yet in contrast to several other biologically important transition metals, finds its primary biological role in its cobalamin form as an enzymatic cofactor for a range of enzymes with unique functions, including methyltransferases and radical isomerases. Although cobalt also acts catalytically in non-cobalamin, cobalt-dependent enzymes such as methionine aminopeptidase. Cobalamin and its derivatives are coronoids, structural units comprised of a cobalt center chelated in tetradentate fashion by a macrocyclic tetrapyrrole corin ring, substituted at many positions with methyl and alkanamide groups. In complete coronoids, a class of coronoids that includes cobalamin, one substituent is a pseudonucleotide moiety terminating in a dimethylbenzimidazole, or DMB, base that back coordinates to the cobalt center intramolecularly in a low-strain 19-membered loop at the lower or alpha axial position. While cobalamin's corin ring closely resembles that of porphyrin rings such as those found in heme, coronoids including cobalamin are distinct from porphyrins in a few key ways. Namely, the corin ligand internal ring is 15-membered compared to porphyrin's 16, the corin ligand is monoanionic whereas a porphyrin ligand is dianionic, and coronoids are not pi conjugated around the entire ring and are non-planar unlike porphyrins. The smaller internal ring size of corin enforces a tight equatorial ligand field, contributing to the low-spin chemistry of cobalamin complexes for cobalt 1, 2, and 3, and additionally, the catalytically relevant cobalt 1 is relatively well stabilized by monoanionic corin, whereas it would be destabilized and inaccessible if coordinated by a dianionic ligand such as porphyrin. Coordinated to the cobalt center at the upper beta position of cobalamin is one of several monodentate ligands such as a cyano, aquo, methyl, or even 5'-deoxy 5'-adenosyl ligands, giving rise to several cobalamin derivatives. The former, cyanocobalamin, is often the derivative present in over-the-counter vitamin B12 supplements, and the latter two, methylcobalamin and adenosylcobalamin, are of especial biological importance as the cofactors used catalytically in methionine synthase and methylmalonyl-CoA mutase due to the unique organometallic reactivity of their cobalt carbon bonds. Methionine synthase catalyzes the methylation of homocysteine to methionine through a catalytically active methylcobalamin prosthetic group, whose axial DMB base has been decoordinated from the cobalt center and replaced by a histidine residue belonging to the methionine synthase protein, a configuration termed base-off, histidine-on. The methyl transfer to homocysteine is generally believed to occur via two successive SN2 reactions conducted by activated thiolate and cobalamin-1 nucleophiles, in what has affectionately been described as a ping-pong mechanism. In the first reaction, methylcobalamin 3's methyl group is abstracted by a zinc-2 activated homocysteine thiolate, yielding methionine and resulting in the reduction of cobalamin 3 to cobalamin 1 via heterolytic cleavage of the cobalt carbon bond. To stabilize the cobalt 1 intermediate, which prefers a tetracoordinate geometry, the axial histidine residue, part of a histidine aspartate serine catalytic triad, is protonated, causing its decoordination from cobalt and yielding a tetracoordinate histidine-off cobalamin 1 complex. Electron paramagnetic resonance has demonstrated that, in contrast to tetracoordinate cobalamin 2, cobalamin 1 appears diamagnetic, suggesting a simple crystal field splitting picture with a filled dz² HOMO. This doubly filled dz² orbital of cobalamin 1 imparts supernucleophilic character to cobalt at the axial position. Then, in the second SN2 reaction, the highly nucleophilic cobalt-1 species abstracts the methyl group from the proton-activated tertiary amine of methyl tetrahydrofolate, producing tetrahydrofolate and oxidizing cobalt-1 back to cobalt-3. Cobalt-3 favors a hexacoordinate geometry and is thus stabilized by the recoordination of the axial histidine. thereby regenerating the hexacoordinate base off histidine on methylcobalamin 3 complex and completing the catalytic cycle. As we have just seen, the action of the histidine aspartate serine triad attenuates the degree of coordination of axial histidine to cobalt, tuning the strength of the organometallic cobalt carbon bond via a so-called thermodynamic trans effect. Put another way, the protonation of histidine by the triad and weakening of axial histidine coordination to cobalt destabilizes cobalt 3 and activates the methyl group for heterolytic abstraction by the thiolate nucleophile to yield cobalt 1. Conversely, deprotonation of histidine and its increased axial coordination favors nucleophilic attack of cobalt 1 on a methyl group and oxidation to cobalt 3. In this way, the triad acts to regulate the histidine on-off state of cobalamin, thereby controlling the organometallic reactivity of the cobalamin cofactor. In effect, methionine synthase keeps homocysteine and methyl tetrahydrofolate levels in check, which otherwise rise precipitously in pernicious anemia, and replenishes methionine and tetrahydrofolate.